hi everyone. My name is Oana. I just started my research group at Genentech, studying cancer single cell tumor dynamics. And I'm really excited to start off our day today thinking about cell state transitions and how to study them using perturbations. Please feel free to ask questions during the talk so that it's more interactive and fun and all that. Okay, so cells are finding themselves in different states. They have to constantly take in signals and process them and decide what to do with their functionality, their morphology, their molecular sort of features. So the way that cells are changing between different states is really at the core of fundamental biological questions that we are all here to study together. For example, how do we go from like a single cell to all the different types of cells in our body and the whole organism? How do cells know where they are and what signals to listen to and how to change in response to those? Of course, how do we create a disease cell state changes, and then how do treatments try to point cells back to some healthy state or sort of things to stop progression? And really the way I think about it is that there's two fundamental questions here. One is identifying what regulates cell states, and hopefully then using this type of knowledge to understand how we can bias cells to specific cell states that would be desired. And of course, knowing what regulates the cell states would be really helpful for understanding biasing cell state decisions and vice versa. And so we can think about what determines cell state transitions. Of course, there are many things internally in the cells. There could be different transcription factors that are driving specific sets of genes. There could be mutations or just stochastic differences between cells. And then there's external factors like cells communicating with each other, cells being in different tissue locations and sort of reflecting that. And again, as I was saying, our goal for at least the next hour is thinking about how we can identify the regulators of such transitions. And one way to do it is observational. So just look at cells in different cell states, look at genes or any other things that we could measure and see which types of regulators are associating with different cell states. So that produces a lot of hypotheses that we would then have to sort of follow up and really study to understand which ones are causally driving the cell states. And a more direct and sort of exciting way is to do that by doing specific perturbations to cells. So for example, if we have a cell that undergoes a specific cell state change, and we want to understand basically this arrow, we want to understand what controls this cell state change. One thing we could do is maybe knock out a gene, and if that cell state change doesn't happen anymore, then conclude that that particular gene we knocked out was necessary for the cell state transition. And similarly, for a different gene, it might be that we are actually creating a cell state. So by activation type of perturbations, we could learn the genes that are sufficient for a cell state transition. And so today we're going to talk about computational approaches for connecting different types of perturbations to the cell state transitions that they induce. And to do that, let's talk a little bit about the experimental ways in which we can do many perturbations on a large scale. So we can do that using pooled CRISPR screens. Essentially, we start with a bunch of cells. We introduce guide RNAs into them that guide the CRISPR system to produce specific knockouts in cells or other types of perturbations. So each cell gets a different guide RNA, so represented here in different colors. And then, depending on exactly what we're interested in studying, we can select for a specific outcome and check whether specific perturbations were enriched or depleted after the selection. So if they were enriched, it means that losing that gene was like basically made cells go to the state more. So that gene was sort of repressing the cell state. And depletion would be the other way around. And so this is great when you know the stuff you're looking for. But there are many cell states that happen in response to perturbations that you might not know a priori could happen. And so what's really exciting these days is thinking about ways to come up with more comprehensive phenotyping approaches where you don't have to sort of know what you're looking for beforehand. And so the way we could think about this is basically stop selecting for a specific outcome and looking at enrichment and depletion, and instead use single-cell RNA sequencing to actually profile the molecular cell state of the cells for each perturbation. And Perturb-Seq is a single-cell genomics technology that allows us to do these types of pooled screens with single-cell readouts, such as gene expression. So again, we start with a pooled library of guide RNAs. We transduce them into cells so that each cell gets a different one. And then we get two things for each cell. So we do single-cell RNA sequencing to get the expression profile of a cell. So that's sort of the proxy for the cell state. And then we do a dial-out PCR to basically fish out the specific guide or the specific perturbation that was present in the cell. And then we get some data like this, where we have a gene expression count matrix. So for each cell, for each column there, we have for each gene a value of its abundance. But then, I mean, that's sort of your typical single-cell gene expression data. Then we also have these labels on the cells that tell us which perturbations they receive. For example, the yellow cells, we all receive perturbation 1, pink perturbation 2, and so on. So then we can take all the cells in our experiment and sort of split them, like label them by what perturbations they have, and then essentially split them into cells that were unperturbed, they didn't get any perturbation, and cells that were perturbed in different ways with different perturbations. And then really our goal is to estimate what is each perturbation doing to the cells. That's what we're going to talk about today. And of course, there are many varieties of different perturbations. So I've talked a lot about knocking out genes, but there are, the variety of things that are sort of summarized in the review I mentioned below. There we can do base editing or more gene regulatory types of perturbations like CRISPR inactivation or CRISPR activation, doing different epigenomic modifications like DNA methylation. And then we can have sort of more arrayed type experiments where we treat cells with different drugs in different sort of wells of a plate, or treat them with different growth factors or secreted factors. And sort of recent advances allow us to combine these together as well into the larger and larger scales, for example, via pipeline. But back to our problem. So let's say we did a bunch of different perturbations in a pool, and we want to estimate the effect of these perturbations. So this is what it would look like sort of in an abstract cell state space. So here's the original unperturbed cell. Maybe the first perturbation kind of takes you up in this space. And maybe the second one does a completely different thing. Maybe the third one is pretty similar to the first one. Maybe some perturbations, they don't do much. So we want today to sort of learn about how to reason in this cell state space in order to basically predict what combinations of perturbations would do, or predict what entirely new perturbations what kind of cell states they could bring about. And of course, this sort of abstract cell state space then has to be brought back to reality and converted into a gene expression profile. And we'll talk about how to do that. But you can imagine there can be quite a bit of nonlinearity between the cell state space and the actual gene expression profile. Like a small change in the cell state space might be like a large change in specific pathways, or it could be reflected, like larger changes could be associated with specific types of pathways. Okay, so that's sort of the intro. Today I'll talk about 3 types of questions that are sort of exciting to answer from perturbational data. This is not an exhaustive list, obviously, of things that we could ask or approaches we could use, but we only have so much time, and chime in with more ideas and okay, there's a question. Why not directly consider the gene expression as the cell state? So we'll talk about that, but so in general, by definition, I think we think of gene expression as a cell state. So maybe this latent space, I shouldn't call it cell state space, and I should just call it latent space. And so the reason we will work in this latent space would be that it's a space where we try to enforce some sort of additivity between perturbations, which might not translate to linear relationships in gene expression space. Yeah, cool. I think this will be answered better when we talk more about the latent space. Other questions? Cool. All right, so let's start simple. How do we actually estimate the effect of a single perturbation? So let's start just even with the effect of a single perturbation on a single gene here, on a single row in our data. So one thing, so the simplest thing we can do is use a linear regression to fit our gene expression across cells as a function of perturbations. So we'll basically have a vector of expression values for the cell, and then we'll have these input data that tell us which cell got perturbation 1. So for example, these 3 got perturbation 1, and so on for each perturbation, like this last one got perturbation n. And then essentially, we are trying to identify these beta coefficients, which tell us whether perturbation basically increases or decreases the expression of a gene. Yes, sorry. Yeah, so this is just like gene. Yeah, so this is basically like the expression level of gene G across. So in a plot, this would look something like this, where on the x-axis we would have, basically like a 0 or 1, whether cell had a perturbation or not. And then essentially we're trying to basically fit a line, and beta will be the slope of that line. I think there was a question.
Yeah, the question is that shouldn't you also add a base level, no perturbation level?
So there will be some cells that don't have any perturbations. So they will be there is also sort of an intercept basically that is going to account for the base level. And depending on how you set that up, that could be the cells that have no perturbation in them, or it could be the cells that have a control perturbation. Without other questions? Okay, cool. Okay, so beta, essentially, if it's positive, it means the perturbation increased the expression of a gene. If it's negative, decreased it. And so we can do this across all the different perturbations. And we basically get for this one gene G, a beta value for the effect of each perturbation. And then we can actually do this, of course, across all of the genes. So we do it for the first gene. For gene 1, we get a vector. Do it for gene 2, gene n, and so on. So at the end of the day, we actually get a matrix, which we call a regulatory matrix, which tells us for each gene how each perturbation affects it. This is what it looks like for an example of knocking out regulators of immune cell function when dendritic cells respond to LPS, in case you are interested. So again, each row here is a gene, each column is a perturbation, and the values are the effect of the perturbation on the gene. So looking at this, you kind of see that some perturbations have similar effects, so they cluster together into these co-functional modules, as we call them, basically perturbations with similar outcomes. And then similarly, genes cluster into gene programs. These are genes that co-vary across perturbations. For example, they're regulated by the same transcription factors, or they're involved in sort of the same types of pathways. So then we can link essentially the modules of perturbations to the gene programs, and basically learn, the groups of what types of perturbations lead to what types of changes in the particular system we study. Okay, so this is going to be a slide that we will build up all the different models we'll talk about. So really our goal today is to sort of learn a mathematical way of taking an unperturbed cell, adding a perturbation to it, and getting the perturbed cell state. So what I've told you so far.
Yes, I imagine you tell us more about the assumptions of this additive model, or assumptions of independence between the perturbation effects and things.
Yeah, well, that's what's coming.
Okay, thank you.
But yes, exactly. So was not planned. But so we start from the unperturbed cell state, want to get to the perturbed one, and I've just told you about this way of just estimating the effect of each perturbation individually. And so if we think about how predictive this is in terms of what combinations of perturbations do, it's not very predictive. Because it can only predict if the two perturbations will sort of have an additive effect. If they do something more, like they're epistatic, or you didn't see any effect with them individually, but there's an effect with them together, you couldn't know that from such a linear model. So we'll start talking about how we can predict the complex effect of combinatorial perturbation. So going back here, I've just told you, so you cannot generalize the sort of combinations of perturbations unless they're additive. The one thing you can do is be like, okay, let's measure some combinations and model that. So you go and do another experiment where you also include combinations of perturbations in the same cell, for example, by increasing the concentration of guides, and then cells by chance will get more guides. And so then you'll have cells that have either perturbation 1 or perturbation 2, or both of the perturbations in the same cell. And then you can add to the linear model this type of interaction term, where in addition to the individual effect that we talked about before, you'll have this interaction effect, which is for cells that have both perturbations, is there a change in gene expression that could not be explained simply from the additive, basically from adding together the effect that perturbation 1 has and adding it with the effect that perturbation 2 has. And so this is an example of what it looks like. We have, I'm going to show you two perturbations here. So each row is a gene, and each column is going to be one of two perturbations or their combination. So, so this is the beta. This is the effect of the single perturbation on all these genes. This is the effect of another perturbation on the same genes. So you can see that these perturbations on some genes, they have similar effects. On some genes, they kind of have opposing ones. And then we can ask what happens when we perturb both of them in the same cell. This is what happens. And then essentially look in our model for the interaction term. So for these bottom perturbations here, the interaction term is 0 or close to 0. So those are cases where the two perturbations are sort of acting pretty independently. There's not a change in addition to just the individual changes. But there are other genes on top of different varieties where the outcome of perturbing both genes is actually quite distinct from what you would expect just if they had independent effects. And so, I don't know, for example, here, for these genes, they go down upon NF-kappa B1 perturbation, they go up in RELA, and then in the interaction, they actually go down even more than you would expect from adding these two together. So I've shown you now that we can add this type of interaction term and actually be able to model combinations of perturbations and their effects on genes. So that's very exciting. We kind of estimate a ground truth of what combinations of perturbations do. You're not very happy, and I understand, because you cannot actually predict things about new combinations of genes that that we haven't already measured. Like this is great for sort of descriptive studies of what we've already measured. But what we really want to do is actually learn some fundamental principles for how cells work and make predictions about which new combinations would give us a cell state that we would like. So this is not able to do that. And of course, it's also not able to predict perturbations that were not even seen at all, even sort of as a single perturbation in our data set. And as you can imagine, we'll talk about ways to actually be able to predict these. So the intuition that you might have at this point is that we should be able to make predictions about combinations of perturbations we haven't measured just by their similarity to perturbations that we did measure. Because genes are sort of not all acting independently of each other in crazy ways, but they actually fall in pathways. So genes that maybe are sort of synergizing with other genes, like if you have a group of genes that are all involved in growth, then maybe perturbing either two of them would have similar effects because they're involved in regulating the same type of process. So let's think about how we can actually encode this type of similarity between perturbations to predict the effects of combinations that we haven't yet measured. Okay, so the intuition is that similar perturbations are likely to have similar genetic interactions. So going back to this again, like latent space that we're going to talk about in a second how to get it. But for now, let's just assume that we have it. So we have our unperturbed cell, and we have perturbation 1, goes up, perturbation 2 in a different direction. So perturbation 1 and 2 together would sort of be the addition of these two perturbations. Now, if we know that perturbation 3 is very similar to perturbation 1, they sort of induce very similar effects individually, you could imagine that adding up perturbation 3 and perturbation 2 should be pretty similar to what happened when we added perturbation 1 and perturbation 2. So this is the type of way where measuring just the individual perturbation 3 could actually be informative about perturbation 3's genetic interactions just by the nature of how similar it is to perturbation 1. And so this type of intuition is what is going to drive the next model that I'm going to talk about. And again, we'll also talk about how to map from this latent space to the actual gene expression profile, which can be quite nonlinear. Okay, so how can we learn this cell state space that also has this super nice mathematical arithmetic situation going on? So, one way is described in this paper, compositional perturbation autoencoders described. So there's basically two steps. The first step, we are going to learn what the cell state, low dimensional cell state is. And the second step, we are going to enforce that we have this arithmetic in the space where similar perturbations, they add together and do all these things. So first, let's talk about how we actually come up with this latent cell state space. So one sort of powerful way to do it is using autoencoders. So this is a way of taking the high dimensional gene expression profile and essentially converting it into a low dimensional space that's quite predictive of the origin. So we start with our gene expression profile of a cell as an input. We want something more low dimensional. So we are going to have an encoder neural network that is going to basically map each gene expression profile to this lower dimensional state. So at this point, you can do that in many different ways, right? There's not really a constraint for how to do it. But the constraint will be that this encoding in low dimensional space should have all the information, or should have a lot of information about the variation across our cells, such that it should be basically sufficient to reconstruct or predict the original gene expression profile that we gave it. And that's going to happen through the second neural network called the decoder, which basically is going to map, is going to do kind of the complement of the encoder. So it's going to map from the latent space to gene expression. So we're doing this with neural networks. If we were to do this just in a linear way, it would be PCA. So in PCA, we take our gene expression profiles and we convert them into a low dimensional representation of PC1, PC2, PC3, and so on. And then we decode it. But here we allow to learn more complicated sort of nonlinear functions of like from gene expression to the latent space and from the latent space to gene expression. It's like a longer question than what I can see here. Margie, can I?
Besides the issue with vanilla autoencoders or PCA requiring a bottleneck layer as representation, and the possibility of having a smaller number of parameters to learn, is there a reason to posit a low dimensional latent space?
So, I mean, I guess I can imagine there are a few reasons, and maybe other people can chime in. So one would be just sort of what we do with PCA. So you would do a little bit of denoising by going into this latent space. There are other constraints in some types of autoencoders where you sort of make this latent space have its different dimensions, try to be as independent as possible from each other, which can be interesting. And I mean, the other reason why it's going to be interesting for us is because we are going to add additional constraints onto this latent space and combine it all together into a single loss function and sort of learn about that. If anyone has other insights on
I don't know which type of autoencoders, but I assume also it's because the latent space helps to be denoised enough because you keep only the important information by compressing the information in lower dimensions and reconstructing, you might end up with something clearer. So it depends if you're using later on the output or just the latent space, because also in most of the autoencoders, you cannot use the latent space. You need the variational autoencoders to be meaningful. But that's the reason that usually the autoencoders employ this dimensionality reduction in the middle, and then they reconstruct.
Oh, thank you. Other questions?
Yeah, just a comment. I think beyond the technical reasons, I guess there's a question of whether it makes biological sense for us to think about cell state space in lower dimensions than gene expression. And I think it kind of does because, well, I mean, you can always measure more and more gene products. So it's unlikely that, that the space is that high dimensional. But that would be my intuition. But it's definitely different to the gene expression dimension. And yeah, so whether it's higher or lower, I can't say.
Yeah. The microphone.
To add to that excellent point, I think there's a lot of redundancies in how gene expression is regulated in a cell, and a lot of genes are co-regulated by the same factors. So in order to prevent overfitting, it's helpful to reduce your gene expression space to a lower dimensional space. Because if you look at gene expression in a cell, yeah, there's so much redundancies in like how much information you get from individual genes by themselves versus something more reductive.
Mm-hmm. Yeah, totally. And there was another question about how many genes we have. So usually what people do is they select for the genes that vary across the cells in the data set. So you usually end up with a few thousand, between 2,000 and 5,000 usually. Depends on the number of cells and so on. Okay, so autoencoders, they give us this latent space. I think once I give you the second step, you will be more into the autoencoders. Okay, so the point is the way we do this is that we try to minimize basically this reconstruction error of predicting the original input from this latent space. So now, how can we have this nice sort of stumming ability in the latent space? So the way we'll do this is that we'll actually decompose our expression profiles of a cell into an unperturbed state and then a change induced by each perturbation. So if we think that we have the perturbed cell state, it's going to be starting from an unperturbed cell state, encoding it into this low dimensional space where we can add the effect of the perturbation and then decode it to get the gene expression. And again, the way this will happen is that it will have to be predictive of the resulting gene expression profile. So let me show you how this model works. So we start with our basically cell state autoencoder. So we're going to take our gene expression profiles, do an autoencoder, and map the cells into a d-dimensional space. Now, what happens that's interesting is that we are going to require that this mapping cannot be predictive of the perturbation. So we are going to add on top of that a discriminator that's going to have to predict from the latent space which perturbation a cell had, and it should not be able to do that. So in that way, in this particular step, we're just learning how to map cells into a cell state, into a latent space, just before the perturbation. And then the second component is that we will also have an encoder for the perturbations that maps to the same d-dimensional space, such that adding the effect of the perturbation in that space is then predictive of the total gene expression profile. So in this way, essentially the perturbation effect will need to be able to predict this difference between the unperturbed state and whatever the actual perturbed gene expression profile is. Does that make sense? So now the key thing to remember here, though, is that while we will have this sort of nice additive arithmetic in this latent space, it doesn't mean that that's going to be the same in the gene expression space. So let me give an example. So we have here our two perturbations, P1 and P2, and their sum, that kind of looks like their linear combination in the latent space. Once the decoder takes each of these three cell states and maps them back to the gene expression space, in this particular case that I invented, they could be additive in the, in the double perturbation. But there may well be a different combination of perturbations, like the same P1, but combined with another different perturbation. And again, in cell state space, we are adding together their effects, but the decoder then converts the perturbed cell state in, like the doubly perturbed cell state, into, a genetic interaction, like a synergy here, where basically this gene expression profile is wildly different than what you would expect from the additive effect. And so it's a little bit taking time to get used to it, because essentially in this latent space, all these operations between perturbations will be, linear combinations, but then it's basically up to the decoder to figure out how to convert different types of the different sort of spaces in the cell state space into these wildly different expression profiles. And indeed, it learns then these types of genetic interactions that are non-additive. So it doesn't just learn additive things because the decoder adds this nonlinearity. So here's an example where this is the truth and this is the prediction. You have control cells, and for a bunch of genes, we have control cells plus one perturbation plus the other one, or the double perturbation. And you can see the double perturbation looks different from the two individual ones, and the model is able to predict that. And presumably it's able to predict that because it saw other perturbations that looked similar to the individual perturbations, but from which it also learned combinations that it saw in the training set. And there, there's a variety of such examples. Okay, so back to our cheat sheets here. So we've talked about the compositional autoencoder, where we are doing this nice arithmetic of perturbations in the latent space, and then decode it into, this nonlinear gene expression profiles. And that is very cool, because we can predict new combinations made out of perturbations we've seen. And of course, this performance is going to sort of scale with how many other combinations you've seen. Like if you haven't seen so many combinations, then you learn similarity between perturbations, but you don't know how to convert that into combinatorial effect. And if you want to predict what will happen with a perturbation that you have not measured anything about, you can't do it here because you wouldn't know how to encode it into your latency. Yeah, and then the reason why this learns is that it basically takes in the similarity between perturbations that allows you to extrapolate between combinations of perturbations similar to what it's seen.
Is the lower dimensions latent state D could be correlated with two biological or molecular functions?
One dimension, possibly.
The D-dimensional latent space, yes.
Trained on the D-dimensional space that they have to be, distinct biological things. The D-dimensional space just has to be predictive of the outcome. So if two pathways in our data set happen to always be covarying, then they're going to probably be part of the same latent space dimension. Yeah. All right, so we've talked about how to predict the complex effects of combinatorial perturbations. And now we're getting really excited, and we want to, I mean, you kind of start to feel that if we could, no.
On the previous slide, just a quick question. If I do choose linear encoders and decoders, and I am doing PCA, does this work anywhere near as well, or?
I would have to double check. I feel like this might be something they tried in the paper. Yeah, let's follow up.
Okay.
I mean, I would imagine it wouldn't work as well, because the point is that the decoder finds these different sort of patches in the cell state that can be quite different. And so that's how it predicts synergy between, like in combination. So I would assume it's going to predict much more sort of additive effects, but I'm not sure.
Can we interpret the matrix elements of learned data as epistatic strength between genes in general?
Yes. Cool. Other questions? Okay, so now we've seen similarity between perturbations helped us make predictions about combinations we hadn't measured. So if you get really greedy, you can imagine that there's other types of similarity between perturbations that don't necessarily have to be determined from our perturbation experiment, like just from what we know about genes in general. And so that would allow us maybe to predict the effects of perturbations we've never even measured, if they're similar to perturbations that we did. So that's what we'll talk about next. So the intuition is specifically for perturbations that knock out genes or that perturb genes, we can use some extra knowledge about similarity between genes that we have from co-expression networks or from pathway, belonging of the genes to make predictions about brand new perturbations. So if we have a perturbation we measured and we know it's in the MAPK pathway, and then there's another perturbation we haven't measured, but still in the same pathway, we might make a similar prediction. That's what we are going to do now. And it's basically going to be really cool. So it's going to be similar to what we did here in that we're going to have a autoencoder for our cell states. So essentially where we're just taking our gene expression profiles and putting them in the low dimensional representation. The thing is, we are going to do the same separately for perturbations. And the way we're going to do that is we're going to come up with some features for perturbations. Again, it might be pathways, it might be co-expression. We'll talk about what they could be. So we'll have an embedding of perturbations that depends on their features, three known features. And so then what we'll do is, given a perturbation, embed the perturbation into the perturbation latent space, then learn another neural that maps between the perturbation latent space and the cell state latent space. And then given that we know where the perturbation takes us in the cell state latent space, use the decoder to get ultimately the gene expression profile. And so you can imagine how basically featurizing perturbations would allow us to make predictions both for new combinations we haven't measured or for perturbations that we have just never seen, as long as we have the same types of features that we use for creating this perturbation embedding. That makes sense? So this is what they do in this paper called Perturb-Net. Would have been kind of cool to call it Perturb-Net. So I've described how this model works, but so the idea is that perturbations are embedded through their features now. So in our model before, they were embedded in such a way that they were predictive of the perturbed gene expression profile. Here, it's just going to depend on their features. And so that means you can come with any new perturbation as long as you have those features and predict cell state changes. Of course, your performance is only going to be so good as sort of the space of perturbations you've seen in your training data, but that's true with any model. And so this is cool because you can have different types of perturbation embeddings for different types of perturbations. So for example, For cyplex, where you do different chemical perturbations, you could just have basically like the chemical structure as a feature of the perturbations. Whereas for Perturb-Seq, you could use more something like gene ontology as a feature. So you get a lot of sort of creativity in how to featureize the perturbations, and that's going to be separate from the autoencoder that learns this latent space for the cell state. And then you put them together and you predict the gene expression profile. There are also sort of other ways of using. Oh, there's a question.
Can a network trained on single and double perturbations predict triple perturbations without ever seeing one?
Yeah, so for this model, it would just have to be like for the 3 perturbations, like as long as you can create some feature that encodes those 3 perturbations together in order to embed them into the perturbation embedding, then you can make a prediction. And similarly, in our previous case where we were doing this arithmetic here, there's no reason why you wouldn't be able to like sum up more perturbations. Again, it depends on your training data, how well you're going to do. I would imagine the more perturbations you sum up, the more it will be important to have seen them in the training set. But that's something one would have to empirically check.
We have another question. Why not force the perturbation embedding space to have the same semantics as the cell state embedding space? The mapping is the identity.
So would tie really nicely the model we talked before with this one. And I'd be very excited to see that happen. So it would be nice because it would be a very interpretable way of looking at the perturbation effects. But instead of linking them back just to the idea of the perturbation, you link them back to the features of perturbation. So stay tuned for sure that will happen or is happening.
The benefit of being able to predict unseen perturbations, does having this like more informative perturbation embedding just yield better performance even on the same task?
Probably. I kind of I mean, in all these papers, they're doing better on some things than other ones. I would have to look more carefully exactly where they do better, but The other thing is that you also have more of this interpretability because you can link back to the features of the perturbations, what kind of clusters your perturbation and predicts the cell states. Okay, maybe I will move on quickly because I don't want to keep you guys over time. There are also other types of ways of featureizing that are more out there. So not operating in cell space at all and just embedding genes and embedding perturbations by their similarity, and then doing some neural network and then predicting the gene expression outputs. We can talk more in the discussion. What I want to leave you now in the last few minutes is just sort of the outlook, where we go from here. The very short-term outlook is get excited for Charlotte's talk. So what happens when we actually, we've talked a lot about the effective perturbations as if they were sort of mean effects, moving cells into a particular direction. But actually, we are measuring a distribution of cells for each perturbation. So modeling that heterogeneity in the response will be very interesting. And optimal transfer is a way to do that. And so Charlotte will talk about it. There's also the cinema OT method. You can check it out. And then there's additional future directions that might be of interest. One is not focusing just on gene expression, but looking at different modalities, chromatin accessibility, imaging, and so on. Taking into account the behavior of cells sort of across time. So taking into account growth rates of cells and sort of effects on fitness, vectories. For all of these types of models, we really need good baseline models to predict again, to address some of the questions about how much better do they do compared to like simple things like PPA. And for that, we also need sort of ever increasing ground truth data sets, especially for perturbations that are large enough and that are sort of diverse enough to to really let us know when a model is doing well. And in that direction, one interesting way to think about it is these types of compressed measurements, and I'm sure being at the Broad, you've heard a lot about it. But you could sort of increase the numbers of perturbations you profile. You can take advantage of the fact that gene expression is sort of structured. So you could measure multiple cells together, and in that way measure more combinations of perturbations in the same experiment, since most of them will be additive. Or you could have multiple perturbations in the same cell, and again, try to disentangle their effect mathematically. You can sort of watch this video on YouTube and then learn more from Aviv. Yeah, and with that, I'll just take any other questions.
Thanks, Oana. That was a really thorough overview of this important area of single cell biology. I was just wondering, when you were kind of annotating the data at the beginning, you introduced this label on each cell, whether it has had a certain perturbation or not. Is there any chance that these labels aren't exactly correct? Is it sometimes the case that I just don't know how these are measured. So could it sometimes be that we think we introduce the perturbation, but it isn't actually inducing an effect on the cell state?
Yeah, for sure. So the way we measure it is that the guide RNA that are guiding the CRISPR system to produce the knockout, like we are basically sequencing them or sequencing some barcodes associated with them. So so there are a few types of errors you could make here. One of them is assuming that a cell has a perturbation that it doesn't have, that could happen due to ambient RNA. And there are ways to sort of like basically set a threshold on how, express the perturbation is in the cell. The other case is when a cell really does have the guide, but it doesn't have the perturbation that you expect. For example, if that perturbation is very deleterious and the cells die, and then the ones that remain sort of have a heterozygous one, or what usually happens is that there's an indel that occurs, or if that indel doesn't change, the frame of the protein, it might be fine. So for those, there are ways to basically compare with control cells and see if the deviation is the one you would expect, and then maybe basically like have a set of high confidence perturbed cells. Focus on those.
It's the first time I'm, let's say, not hearing about it, but seeing such an in-depth overview. Are there works on how to use this to connect it to networks? It seems like a direct link between the two.
Yeah, so honestly, that is the part that's missing here because there's only so much time. But there are parallel efforts basically taking these perturbations and trying to derive basically causal networks or like Bayesian networks that are predicting what each gene is doing and how it's affecting other genes. Because you can imagine, like, if you're perturbing a transcription factor and it's target genes, you can kind of basically group things. And ultimately, if you really want to generalize to something new, that's the way to go. So yeah, there's a lot of literature and it's blooming at this moment.
I was interested in the like modules and gene programs. And so like, if you think about gene expression programs from the perspective of like, principal components, how do the programs that are here in terms of genes correlated across perturbation, how do you interpret those biologically in a way that's different to kind of genes that are just correlated?
Sure, you can interpret it very much unless you have either other information or more perturbations that to kind of group genes further. Guess we can talk more in the discussion, but because I don't know if I understood your question.
We have more questions in the chat also. What are the major pitfalls we should keep in mind? Is it mainly overfitting? What about sensitivity?
So I think overfitting is it's overfitting in a particular way. So I think basically going too far away from what we've seen in training is going to degrade the results very much. So for example, the CPA, the second model we talked about, they actually compute a measure of how similar is the combinatorial perturbation to what we've seen in training to kind of give you a measure of their uncertainty in their perturbation prediction. Other pitfalls. I think we need also just to have much more sort of replicate experiments of the same perturbations and then understand the noise structure much better. A lot of the work is sort of focusing on, we did one big experiment with a lot of perturbations and seeing exactly the way that they replicate, in my experience, is a problem of its own too.
You deal with doublets.
You deal with them similarly to any other sort of single cell works. You can look for, I guess it's a little bit harder because when you have different cell types, you can identify doublets as cells that have basically markers of different types of cells or different types of clusters. In perturbations, the effect sizes are much smaller, so the differences between different perturbations are smaller. So it's hard to basically identify which cells are doublets just by looking at their expression. So what people tend to do is like look for the clusters of cells that have sort of are enriched for more perturbations, and see sort of that the analysis is robust when you leave those out and so on. But we definitely need more methods to do that.
Hi, Oana. Thank you so much for the talk. It was a great presentation. I have a question regarding the validation of those predictions. So we're talking about predicting combinations of perturbations, and I wonder what's the the way that the community is thinking about performance metrics or maybe experiments to validate that those predictions make sense.
Yeah, so I think that's also sort of an ongoing field. Maybe Charlotte will also talk more about it. I think like the key way it has been done so far is to look at how predictive the model is for some perturbations that were held out from training. The next best thing would be like sort of predict like a separate experiment or like in a different species or a different system and so on. And the metrics to sort of estimate how well you're doing will have to be a combination of these, like the prediction accuracy, maybe the interpretability. And there are then other ways to sort of look at, for example, genetic interactions and compare it to protein complexes and assume that proteins in the same complex should have genetic interactions. So there's many ways where there's sort of there should be a bias in that direction, but not clear which of these should be genetic interactions. Yeah.
Great, thank you.
So another question is about the opportunities of using transfer learning. What do you think about that? Like
using what?
Transfer learning, like using.
Transfer learning.
Yeah, so we have many large cell atlases nowadays, and people have, learned nice cell state models there. So is it possible to have some transfer learning to help a better cell model for perturbation, probably improving prediction on unseen cell types?
Yeah, I think that would be very interesting. So I think these large atlases could, like in the last model, they could have like a really solid sort of gene expression latent space. And one could try to map the perturbations to these sort of ever increasing cell latent spaces. I think the other thing is that there's going to be more and more genome-wide or really large screens, and comparing the same perturbation across different contexts, like different types of cells or different species, will be interesting. So in that case, a model like the second one, which decomposes the sort of unperturbed state, which could be, like different cell types, different species, from the effect of the perturbation, which might be more consistent, would be interesting. And maybe there one would have to also have an interaction term of, well, in this species, you tend to also have these weird genetic interactions that you don't have in the other one. Yeah, it will be interesting.
I think the next question is relevant somehow to what you said. That like for both regression model and encoder, is it possible to use a model that is trained on one unperturbed state to predict another unperturbed state? Maybe cell type is an example.
Yeah, so there are papers that do this. One of them is SCGen, exactly what you said.
Welcome to my talk, and thanks a lot, Oana, for introducing most of what I'm going to speak about today. So I'm going to focus on something we call neural optimal transport methods for predicting single-cell responses to perturbations. And the setup is very similar to what Oana has already introduced. So we have a set of single cells in a fairly high-dimensional data space, which are measured with the techniques she introduced. And this can be a tumor sample of a patient, whatnot. It can also be just like mixture of certain cell lines. And we assume that these cells are going to change upon perturbation, for example, cancer drugs. And we would like to predict the outcome of the treatment really on the level of single cells and really predict the perturbed state of each cell in this very high dimensional data space. And we would like to learn this with a neural network, because if we have a new patient coming into the clinic, potentially we would like to understand how does this particular patient respond to the treatment, which we have, like, using a model we have trained on a previous patient cohort. The issue is, and I think this is what she mentioned as well, is that every time we measure a cell, we need to destroy it. Meaning, I can measure cells which are unperturbed, I can measure cells which are perturbed, but this will be two distinct sets of cells. And these are unpaired, meaning I have basically two distributions, or like kind of samples of a distribution of unperturbed and perturbed cells, but no alignment between them, because these are in fact different cells. I just assume that a cell of a very similar cell state is in the sample after the perturbation, upon perturbation, and I can find some correspondence, and finding this correspondence, in fact, helps me learning out my function F. And so now I would like to predict for an X, this matching matrix, or like kind of reconstruct this alignment to predict the perturbed state of a cell T of X. And we seek a parametrized map T star, so an optimal map T star that transforms, because we have to operate on the level of distributions, as we do not have trajectories like the same cell unperturbed and perturbed. We want to have a mapping which transforms a distribution mu into the distribution upon perturbation mu, such that we have like certain criterions. So this push forward map needs to somehow push all cells, no cells can get lost on the way. But what is the optimal T? So ideally, we are looking for a cell which is very similar upon perturbation. So we want to match a cell to its closest somehow cell in the next, to the closest cell in the next point, like after perturbation, using some form of distance metric. And then we would like to find an overall matching, which, even though there might be some misalignments, finds on average the matching connected to the lowest somehow discrepancy cost. And of course, we still would like to have a push forward map, which somehow pushes entire mu on nu. And this in the end is a result, or like kind of a concept which is very famous, which is optimal transport. So we try to find an optimal transport map such that the overall transport cost is low, meaning the matching which describes the cellular shift from unperturbed to perturbed is the one corresponding to the smallest transport cost. And so this is what we are going to do today. So we have cells unperturbed. We would like to predict the perturbed states for different drugs, drugs in combination, and so on and so forth. And I'm going to start my talk with the work where we just like really learn this mapping. I will follow up with some extensions to maybe learn a generalized map, which can be conditioned on all types of drugs or whatever context. We also need to incorporate and account for the fact that, of course, cells are going to die and cells are growing. I'll continue with a work where we have a slightly more challenging setup, where we not only have perturbed and unperturbed states, but we in fact actually have multiple snapshots we are observing, and we rather want to learn the flow in between. And then I will just give a sneak peek of like what types of methods might be exciting in future, which are potentially even more powerful than the ones we are looking at. So the goal is now to learn this Morse map for a particular drug. And so this Morse map is an optimal transport map. Morse is a guy who did optimal transport research when Napoleon was still alive. And the nice thing about this Morse map is that there are certain theoretical guarantees which come in handy. So for example, this existence of is guaranteed under fairly general conditions. And most importantly, for a certain cost function, the cost function L2, there's a famous theorem by Bunne, which states that there must exist a convex potential F such that this Morse map is given by the gradient of the convex function. So this might sound very abstract, but the only thing we need to remember is somehow it's sufficient to somehow find a convex potential, like a convex function F, a function which is convex with respect to its inputs. And this allows us to recover the Morse map, the push-forward operator between our unperturbed and perturbed states. How do we learn? Because now, instead of parameterizing a Morse map, like a rather complex mapping, we just need to learn a function, a convex function. And in fact, we can use a network architecture known as input convex neural networks, proposed by Brendan Amos in 2017, to parameterize this function class. So what are input convex neural networks? So usually in neural networks, we just have a simple input X. For input convex neural networks, we always keep track of our previous hidden representation. So now we initialize our previous hidden representation to 0. And now in a classical neural network, we just multiply our input by some weight matrix and add a bias term, pass this to an activation function and get the first hidden representation. And usually we would just continue this, right? In a classical standard neural network, we would just multiply our previous hidden representation Z1 with the weight matrix W1. But input convex neural networks, we always feed in back the inputs. So we always have basically like a second path, which is continuously passed in and multiplied with the previous hidden representation. And in order to actually be a convex neural network, meaning input convex with respect to X, all weight matrices which depend on the previous hidden representations need to be positive, and the activation functions need to be increasing convex. So now what do we have? We have a setup where we can learn the Morse map through a convex function. We can parameterize the convex function via a neural network class, which parameterizes like the family of convex functions. And we are not alone doing this. So there has been a large body of work which recently has discovered this way of learning OT, starting with Makuva in 2020. And this is something we heavily use in the first part of this work, but also by Alexander Korotin at Skoltech, David Alvarez-Melis in Microsoft, and even Brendan Amos now also at Facebook. Meta. So we do one more approximation step. So before we had this sup over one convex function, and we had the dependency on this conjugate, which is hard to approximate. So we just do one slight modification by approximating this conjugate by another convex function. And we have now our final objective, which is a min-max scheme over two convex potentials, which we both parameterize with two independent convex neural networks. And we can train this in a very similar fashion as historically Wasserstein-GANs have been trained. In fact, Wasserstein-GANs are very, very similar, just relying on a different cost function, L1 and not L2. And so now we have a parameterization of our Morse map, and using the gradient of a convex function, we are able to map back and forth. So from the unperturbed state, the gradient of the first convex function, the first dual potential, pushes us into the perturbed state and the other one back. And so this is what we do in CELOPS, and let's see why this is beneficial. So just remember, we start with two distributions, unperturbed and perturbed, which we call control and treated. And we look at different types of data. The first one is microscopy based. This is a technology for eye, which is able to iteratively stain and not only measure certain morphology features by extracting the properties from the microscope image itself, but also look at certain expression levels of PERG inside the entire cell or other protein signaling within the cell nucleus only. But then, of course, also, and this is what mostly was covered in Oana's talk, we of course also look at single-cell RNA sequencing data, where we track up to 1,500 or even more genes. So let's start with a very simple setup. So we have in the first, just projected down to the first principal component, the control and the treated population. And this here is a mixture of two cell lines, where we know one cell line is actually responding and the other one is not. And this is exactly what we can see. We see one seems to slightly shift, while the other one stays fairly stationary. How can we look at this? So we always look at this on the level of distributions. So this is somehow like these two measures, which we want to, like the push in these two measures, which we would try to learn. And the easiest way to do so is just do a linear shift in the data space. Meaning we can somehow describe cellular behavior by just like modeling the average response and adding the average response to each individual cell. What we can see is that the fit is not terrible, right? So on average, the behavior is met, but we are not able to somehow learn that the response of that one cell line is not responding well. The other method is like of the class of autoencoders, which Oana introduced. And one challenge of autoencoders is while they don't need to have so they assume a linear shift in a latent space. So overall, could capture nonlinear behavior. It's often very, very tricky to find a perfect latent space in which this hypothesis of the linear shift is actually provided. Meaning that even here in certain setups, sometimes better, sometimes worse, we are not able to really like capture the fine-grained response of these two populations. So these are the baselines we are mostly looking at. And what does cellular do in fact? Like cellular doesn't make any assumption on how this perturbation shift looks like. The only assumption we make is that we can somehow reconstruct this function f describing the perturbation effect based on learning a matching or like kind of learning the structure, which assumes that we can find the corresponding cell types, which is similar to the previous one, like this minimal transport hypothesis. And this allows us to now just learn somehow this potential field, which moves part of the space quite far. So one cell line is pushed quite extensively, while the other one stays mostly stationary.
A question about the evaluation. So I guess you have your method where you're approximating the map by the gradient of these convex, input convex neural networks. Another option would be to introduce a regularization term in some kind of adversarial approach like you're mentioning.
Yes.
So could you provide some intuition for why one might favor the input convex approach versus the...
No, in fact, so when we started, we somehow assumed that you need to have an optimal transport hypothesis, which is very, very strict. And these input convex neural networks, they really somehow strictly fit this OT hypothesis. And this, in fact, might not be true. So it might be also possible to just have a rather unregularized neural network, in which you enforce certain properties via regularizers. So there's alternatives. Basically, it's just the first shift we wanted to make is somehow not enforce the nature of the shift in the first place. And we really strictly enforce the OT hypothesis, which might make sense when this is perfectly met, but probably something in between or something even further might be an even better answer. Yeah.
And my second question was about here, the hypothesis is that the perturbed cell is similar to the unperturbed cell. Would this approach also work in the case when you wanted to do Gromov-Wasserstein, where the distances between pairs of cells are similar between perturbed and unperturbed?
Yeah, so the problem with Gromov-Wasserstein, so basically the way we approach this is always via some form of dual, which we do not have in the setup of Gromov-Wasserstein. But the Gromov-Wasserstein allows us to basically match distributions which lie in incomparable spaces, so different dimensions or different data types, and this is often used for somehow integrating different modalities into the training. I think there are some initial endeavors on finding some neural approximations for Gromov-Wasserstein, but I think the main issue is here that you often have to go via the primal problem, which is prone to be rather unstable. So one thing which is important to note, the fact, and this we will also see later, the fact that we map for and back the cycle consistency actually increases quite some stability into the training. So there's also been other work which somehow tries to, mainly by Karen Young, former PhD student of Caroline Uhler. She has approached this, developed very nice techniques, kind of going by the primal. The difficulty is somehow the stability during training.
I have a pretty naive question, so if I don't want to hold this back, but from when you were describing the neural networks, I kind of wanted to ask again about what's the relationship between adding the input again and the optimal transport. How does that?
Yeah. So basically, the neural network structure itself is somehow independent of the OT. So the only thing this neural networks, so this is independent work, like Brandon Amos introduced these input convex neural networks in actually a very, very different setup, more like the control theory reinforcement learning world, where you have you want to somehow learn the solution to an optimization problem. And it's nice to have this property. It's just a neural network architecture which guarantees that the overall architecture, the output basically is input, it's input convex, like it's convex with respect to the input. And so there's some artifact why you always feed in basically back the input. To be honest, my super thorough understanding of ICNN is why they are built this way. I actually don't have it. There might be other ways of actually constructing neural networks which approximate the family of convex functions. Thank you.
What is the intuition for computing optimal transport on cells between two cell types? Is it possible that the real transformation is local between cell states, so it doesn't need the global minimal transport?
Yeah, I mean, so generally it's, to a certain degree, it's some form of nearest neighbor approximation, right? But just like optimal transport gives us a way of like, on average for all cells, find the matching which is somehow best for this type of setup. So, but then similar cell types ideally are mapped, like the cell types are mapped to the same cell type after perturbation. So the idea of here using OT, and we are also not the first doing this, so there has been work by Jeffrey Schiebinger already in 2019 in Cell, using optimal transport for learning developmental trajectories. It's really that it provides us with a good matching, because it's the matching overall which finds cells which are close to each other. So this is somehow why we use OTS. And I mean, the biggest problem here is that we never have a ground truth, right? So we never have actually a ground truth, what is the perturbed state of a unperturbed cell? I mean, we might have with like the rise of methods like LifeSeq, but still not on the scale which we somehow need to actually learn these functions. And so all the approaches on the next couple of slides is us trying to understand if what we learn makes sense. And one easy way of doing so is just measuring the distribution, the distributional distance of what I predict and what I actually see. So I'm taking the MMD, this is one distributional metric, Wasserstein metric is another one, or just simply the somehow drug score, the average Euclidean distance between the outputs I predict and the treated cells I actually observe. And what we can see is that also with these distributional methods, cellular learns fairly accurately. And if we zoom into two features out of the like 60 features we have in this particular data set, we have, for example, for PAKT and PERK, the marginals of the control population and then the treated population. And what we would like to learn is a method which shifts this light blue curve into the dark blue curve. So the average, as you can see, really fits the average. And so do baselines such as estrogen. They are a bit more accurate often than the average, and they also work much better on more high dimensional data sets. So this is a fairly low one. But still, they more like, because they assume this linear shift, they have difficulties in somehow modeling the heterogeneity of the population, but they are very, very strong in modeling the average effect or the average correlation structure. And CELOT instead, we do not make any assumption on somehow how this shift is going to look like. So it's very easy to also, and because we are doing a matching of the entire population, very easy also to match the cell population in its entire heterogeneity.
How do you compare to if you were to like solve the optimal transport using Synchron or something like?
Yeah, so then because this is always so we thought about this. So ideal, like on the trained set, this should be perfect, but on the test set it doesn't work anymore because it doesn't. So this is always done on the test set. So we have only we only use control cells we never saw, and I mean discrete OT only works basically on I can only do this on the trained part of the control cell. But if you look at the trained part of the control cells, it's perfect.
You could potentially like fit some regression map on the
Yeah,
yes.
Yeah. But this is in the end, I mean, basically we also only derive a method for learning a neural version of OT, basically. There might be other versions. So the problem is we also tried it via the primal OT problem. It's just very unstable. It's really hard to get it running.
Since the advection that OT tries to learn comes from a convex potential, it won't capture barrier hopping phenomena, which may be relevant because we think each cell state as a stable minimum and cell state transition as activation over barrier, or cells with cycling or oscillatory gene expression, because OT's vector field will be curl-free, being a gradient of potential. Shall we worry about these issues?
So there are certain cellular phenomena we cannot. So the entire hypothesis of OT is that I can match them based on finding the closest, like somehow the matching, which conserves the entire correlation structure. And this we also see, and we have run experiments, if we observe a perturbation way further in time, and a lot of things have happened, we are not able to reconstruct this, in particular not with OT. So OT really worked on the basis that somehow close in time we take another measurement. But neither the baselines. If there's some form of oscillatory behavior in between, there's no way of reconstructing this from two somehow static snapshots. This is just the constraint of the problem is very challenging. But yeah, so what we did, and here we can also see just on different types of data sets. So this is a melanoma cell line mixture, which we'll also see on the next slide. Here we have 47 different samples. So there's also a lot of batch effects, which we had to take care of, also for like on average for 35 treatments. So these are both for eye data sets. The challenge, of course, with high dimensional data sets such as gene expression is that even like we also require some form of lower dimensional embedding, because we assume currently that our loss function is L2 in order to have all these theoretical properties. But this means that we cannot like operate directly on like a 1,500 dimensional feature space. It doesn't really matter what type of embedding you use. So you can work with PCA embeddings or some autoencoder. The only requirement we have is that we are able to like map in and out, so that we can conduct the evaluation itself basically on the decoded gene expression space. And here we can see again the average behavior versus SCGen and CAE2 autoencoder. So what we did here is that we had a mixture of two cell lines, melanoma cell lines, where we somehow know, or in particular, Gabriele Gut and Lukas Pelkmans, so the two biological collaborators I worked with, they somehow know how these cell lines are supposed to respond. And we trained this entire thing for 34 drugs. And what we can see, if we just look at the somehow UMAP embedding of the treated space, is that they are all, like every color here, every point is colored with a different treatment, and they all seem to be somehow intermerged. It's very hard to distinguish between the treatments on the two cell lines. If we, however, have now this mapping, we can take a predicted cell, its corresponding control value, subtract it, and get somehow this like treatment response itself. If we embed the treatment response, we can see that all these drugs have a somehow very distinct mechanism of action. And this is very nice. And then we can run downstream analysis, such as the two cell lines, how do they respond? How does each individual feature change? And we can see, certain cell lines respond to certain drugs and the other one does not. And there's more in the paper. We also run some experiments from developmental, like kind of where we have like a developmental trajectory, a data set by Weinreb et al., where we have observations after day 2, 4, and 6. And we have two different subpopulations, one being multipotent and one being oligopotent cells. So we have cells of higher and lower potency. And the question we ask is, okay, so if you train on both, this is nice, but can we somehow generalize? Can we take, for example, the oligopotent transport map, only trained on oligopotent, and make predictions on multipotent, or vice versa? And what we can see is that with cells of higher potency, so training on multipotent and then predicting something on oligopotent, we can actually make reasonable predictions. but the other way around, we cannot. And last but not least, we looked at an initial study of having a patient cohort, training on a subset of patients, learning like one perturbation model. This is a data set by Kang et al. And it has a set of 8 lupus patients. And we try to predict basically now the treatment using this generalized perturbation model on a patient, meaning we have incoming control cells, we take this generalized perturbation model, and we predict the treatment effect. The problem here that this is a very somehow tricky task, because on the one hand, we want to learn a coherent and robust model, allowing for treatment suggestions, but we want to be also very sure that this makes sense. But we don't want to just learn the population average. We want to somehow stay and make personalized treatment decisions. And the results is that, so unfortunately, this data set is fairly homogeneous. We actually were expecting the task to be much more challenging, but the patients are very, very similar. And if we predict and somehow hold out patients, there is a slight, like somehow decrease, but it's actually not that dramatic, because the patients are in fact very, very similar. You do not have the same if you look at cancer patients. Currently, we are running a giant study where we have 47 melanoma patients and microscopy images of each of them for multiple treatments run in parallel. There we have heterogeneous cancer responses, and things are much more challenging. So what we observed in particular when working with the patients, that a pair of perturbed and unperturbed cells never arise in isolation, meaning they're always associated to some context. This could be, I have different subpopulations and I have additional meta-information. I know that this is a patient who has stage 4 whatever cancer. And of course, this is very important. Which driver mutation does he have? And these are all things I can measure, which might not be included in my features itself, but which might give me an informed idea of, because they respond differently, I would like to learn different models for each subpopulation. And because sometimes it's very hard to distinguish really subpopulations well, ideally, I would like just to feed in all meta information I have in my transport model and somehow understand how does the Morse map look like given this particular context. Or if we are thinking a bit further, we can also say, why do we actually learn an OT model for each individual drug? Instead, why don't we feed in the drug just as a condition and then potentially also condition on a set of drugs, like a combination treatment? And so for this, we developed Condor, which we are presenting this year, so later in December. And this is now the task is to learn a family of Morse maps, which for each particular context returns the corresponding Morse map associated to the context, which merges this corresponding mu I to the perturbed state mu I. And before, we just had a function which took as input the cell. Now we have a function which needs to take the cell as well as an additional context vector. And we stay somehow in the assumption that we strictly want to parameterize this as an OT. We can, of course, lift this assumption, but now it only needs to be partially convex, this function. It needs to be convex with respect to the input cell, but not with respect to the context. And this can be seen somehow as a hypernetwork, like a multitask setting where we learn several tasks at once. And the network structure we learn needs to somehow return a particular parameterization for each individual context.
Medium of
yes, that was the gradient of this. Yeah. So it's still like it's a function which takes as inputs 2, and the function will be input convex with the cell. But in the end, the Morse map is the gradient of this F with respect to the cell.
Charlotte, can you remind us what was the meaning of the context? And is it always taking on discrete values, or could it ever be a gradient?
Yes, so I'll talk about this in the next slide, the second next slide. Yeah. Just to briefly understand what are these partially input convex neural networks, but because it's also not something we came up ourselves, but we found, in fact, 2 years later or a year later in the same paper by Brendan. So now we again have our 2 paths. One being the one dependent on the hidden representation, one on the input itself. But now we have a third path, which always feeds in the context and which somehow conditions the neural network structure at each layer. And so this is what we are going to use. So now the aim is to learn a global family of context-aware Morse maps. And the context can be very, very different, right? Like as Alex said, so this can be a simple scalar because I just condition on the time or the dosage level. It can be something much more complicated, but still easy because we just have a vector which includes metadata or some patient identifier. And last but not least, it can be something very abstract, such as an action in a reinforcement learning setup or a perturbation. And now it's much more challenging. How do I encode left-right into the neural network? I need to have some form of embedding. And so this is why we derived, and this is something which is still, I mean, this is published in Europe, but like hasn't, like, so it's still like somehow in the machine learning community and it needs to make the transition to the bio community. But for now, we just like derived a fairly general architecture consisting of 3 parts, the partial ICNN, which still parameterizes our Morse map, which takes in 2 inputs, the first one just being the measure, the second one being the context. For the context, we need some form of word embedding, or some form of embedding. This can be something simple as a word, one hot. The problem is we cannot generalize to unseen drugs potentially, if this is something we want, but we can very easily model combinations by just having a multi-hot. But it can also be something more abstract. I want to have some form of drug embedding, perturbation embedding, and this is work Oana was introducing as well. People have done so. And this is something you can use here as well, where similar drugs are encoded close by in this like embedding space, such that if I have a new drug, I know the location in the space, I can somehow generalize to this new unseen drug. In such cases, we of course need to also have much more complicated combinators because we do not know if these effects are additive or actually cancel each other out and so on and so forth. So this is something we need to learn as well. And so now we tested it Perturb-Seq data set using the methods Oana introduced. And this is a data set from Norman et al. Published in 2019, just on top of, like, I think it's a follow-up work to this initial work of Dixit. And we have genetic perturbations and so genetic knockouts, which we pass into the embedding module, combine to a joint embedding, and then pass to the Morse map as well as passing the input samples. And we compare this against this computational perturbation, compositional perturbation autoencoder by Lotafali et al., so from the TICE group. And so this was somehow the first and actually only method which really, at this point where we were looking at it, modeled composition, like combinations of perturbations. And this is just a very simple graph. Every time it's a Wasserstein plot, we compare against some, how close are we in predicting the distribution of perturbed states. And every time we are above the line, our method is slightly better. And what we can see, of course, there's a decrease when predicting combinations. And here you can just see a very simple UMAP embedding where we just embed jointly the data as well as the perturbed observed data and the predictions of the perturbed states. And what you would like that this is somehow matching quite well. And this is just for one particular combination here, map 2K6, KLF1. But we have, of course, a lot, and the paper has much more. And somehow we often see that CPA, because it has still a very strict assumption on the latent space, is not able to really match the heterogeneity of the population as a whole, but somehow always leaves out certain subpopulations. And we did not do any further analysis, so I do not know why it did not match particularly this subpopulation.
I'd be really interested to know if, so you do better in terms of Wasserstein loss than TPA in like basically all of the combinations.
Yeah.
If you were to just look at, for example, in terms of the means of the distribution, do you where are you gaining relative to CPA? Is it really in the tails in the distribution, or is it even in the mean that you're benefiting?
So we also look at the mean, and so this is what I call always perturbation score, but it's de facto the mean. So you look at the average, like the distance between the average profiles. And so in terms of mean, we also match the mean well, but there's like, if you look in particular, like it's much, like CPA is much better if you just look at the mean, but it's not much better than it's comparable. Same for the correlation structure. So we didn't do this so far because we didn't really care about correlation structure, because the task was really to match the heterogeneity, but also if you just evaluate in terms of correlation structure, it's also so it's just, I think it comes a bit with the hypothesis thing, which is made on the like somehow having a linear shift in the latent space. I think you just lose something.
Question: is there something about the like subpopulations that CPA leaves out? Like, do they look less perturbed, or they just happen to be less close to the mean and then they kind of get ignored?
Yeah, I don't know. So I spoke a bit. The authors of CPA. There's also a V2 of CPA. I think it is also. I mean, it has a lot of components, right? So it's very difficult. It has an embedding of the drugs. It has some adversaries. It has the autocoder itself. So it's very difficult to understand somehow where, which part of the model is somehow the problem. Here, the comparison done is with the same type of one-hot encoding. So this is a comparison just using one-hot embedding, same as this is also what CPA is doing. Then CPA is also additionally embedding the certain covariates such as dosage, but otherwise it's very, very similar from the setup. Yeah. And so, of course, if you look at just straight out using CELOT or like ICN-NOT, then of course, because they don't do any conditioning, this is basically if you just learn on average heterogeneous responses. So of course, cells grow and die, and we need to somehow account for this. And so far we've completely ignored it, and so did the other baselines. So usually a coupling tells us, given a point X, where with a certain amount of mass, and so far we always just assume equal mass for all cells, and we continue to do so. How can we predict, or like what is the corresponding Y, or like where is it mapped to, which Y with the corresponding mass M? And so this is of course difficult if we have cell death, because if we have cell death, we do not see apoptotic cells because they are often not measurable. So we just have a quite unbalanced problem. So we have an unbalanced optimal transport problem. And if cells are going to grow, right, then we also have an increase in another part of the space where we have a high level of proliferations. And so now we need to somehow account for the fact that our problem is not balanced anymore. And we have cells which we would push through the map, but which are not even present anymore in the data because we cannot measure them. And how we do this is we look at a notion introduced by Lenaik Shiza at EPFL, the so-called semi-couplings, which somehow divide this coupling into two parts. The first part tells me how much mass is leaving from X. And the second part of the coupling is telling me how much mass is actually arriving. And this allows me to then say, for those cells which have died, no mass is in fact arriving. And so this, in a setup where we say, okay, so we have actually growth, then this would mean that we have duplication. So meaning like starting from a mass 1, coupling 1, gamma 1 tells us there's at point X, something with mass 1 leaving. Coupling gamma 2 tells me there's something arriving with mass 2. And of course, for cell death, this would be then something like 0. And so we do train this because it's very difficult to parameterize these semi-couplings directly. We do so by introducing, somehow by breaking the problem into an unbalanced and a balanced subproblem, where we have a balanced morph map in between. And somehow these semi-couplings are now parameterized via a weighting function, eta and zeta, and the morph map origin. This is something currently in review, and so the balanced morph map itself is still parameterized or learned very similar to what we've seen before. So let's just focus on how do we actually learn these scaling functions, these reweighting functions. And one crucial observation is that if I'm predicting cells, right, I'm pushing forward my previous population here, both populations A and B, through the map, even though these might be dying. But neural networks always have some form of fixed input-output, right? So in a sense, I'm still predicting the perturbed states of cells which are actually supposed to, which I do not see in my data anymore. Now, if I sample a mini batch, what would I sample? I would sample some cells only from distribution A, potentially more, because there might be some level of of proliferation. And now I can compute an unbalanced OT coupling, so just a discrete optimal transport coupling. And this will tell me that because it maps similar cells with similar cells in the data, it's going to tell me the coupling is only in this like upper part of this transport map. And if I sum now over the rows, for those cells which are just perturbed, I'm getting on average, like I will sum to something like a weight of 1. For those cells which have died, there's no mass transported here, so it sums to 0. And for those cells which have grown, it's larger than 1, so somehow close to 2. And so this is now the value, this summing over the rows of this coupling, over this map, allows us to somehow learn another neural network, eta, which tries to match now the sum over these rows. And eta and zeta are then functions which map from a space, from our space to like a single value in R, like a weight for both perturbed and unperturbed states. So here's there's only one work which somehow tries to assess this problem, and this is dependent on which is by Young and Euler, called NUBOT, and called UBOT-GAN, or we called it UBOT-GAN. And here we have a very simple setup just for illustration, where we have two Gaussians in gray, and they are just like moving a bit away. And now what we are predicting is we are still predicting all, and what we see is that we have different weights. So this population is dying, this population is only dying a bit, and this population is actually growing. And of course, when we predict, we still predict somehow all cells, but we also predict the weights, the associated weights, and we know, the population on the bottom is duplicating, some population is really dying, have heavy level of cell death, and so on and so forth. And this is very similar to the approach by Karen Young and Caroline Euler. They also learn somehow weights. They just assess everything from the primate problem, where you have a bit the difficult situation of trading off the cost of transport and the cost of reweighting. And the nice setup here is that we are again left with our data set where we have two discrete subpopulations. One is MIL-A positive, the other one is SOX9 positive. And we have data after 8 and after 24 hours. And if we zoom in here, we can actually see the overall cell change, the change in cell counts for different drugs. For example, olixertinib, after 8 hours, actually shows some level of cell growth, while after 24 hours, it actually shows death only. And now what we can do is that we want, of course, the weights we predict to perfectly correlate with the somehow cell count which we observe in two different subpopulations. And so this is what we do. So here we can see just the correlation of the weights and the cell count fraction for two different subpopulations after 8 hours and after 24 hours. And here we also summon the same problem which was asked by one listener or in the audience. After 24 hours, a lot of things have happened. Some drugs are very, very aggressive. Staurosporin is basically killing everything it sees, right? We have very little cells left, and it's very hard to really like do a good approximation. And even then, it's very difficult to really predict well the correlation. So this is a bit the challenge here. So I think I'm slowly getting to the end of my talk, but so what if we have time series data? So here we have several snapshots and we still make the same assumptions. Somehow cells evolve according to a stochastic process. They don't drastically change too much, so I can remap them based on some form of optimal transport. And so this is now the task is to learn complex dynamics over the entire series, introduce temporal dependencies, and learn some generalized continuous time trajectories of these populations. And if we look at the main assumption we make here is that we can learn these or describe the dynamics of cells by minimizing some form of potential function J. So this is the one hypothesis we make. And then the easiest setting is to just say, okay, I can describe in Euclidean space the vector at the next point in time by just following the negative gradient direction. This is it's well known to be very dependent on the step size we choose, so tau, and it has certain properties of J. It's known to be somehow unstable. Instead, we can look at the proximal scheme, where we say, I want to, over the entire space or over all vectors, potential vectors X, I want to have the one which minimizes my potential because this is what I'm assuming here. And I want to stay close in space. We are, of course, always operating on distributions because we do not have the same unperturbed and perturbed state of a cell. And now we have just a potential function taking as input a measure. We always need to operate on the level of distributions. And we make one simplifying assumption is that we don't have particle interaction. So this is just the sum of the energy of each part. And we can look at a similar scheme, the forward scheme, or we look at the backward scheme, where basically the argument is again minimizing a potential function and staying close in space, now with respect to the Wasserstein metric, because we are in the space of measures. And this is a very famous result called JKO flows by Jordan, Kinderlehrer, and Otto in 1998. It's actually one of the most downloaded papers of the annals of applied mathematics. And so now this is what we derive. So we somehow assume, okay, we learn this free energy functional via a neural network. And then given a starting point ρ0, we predict the population at the next point in time by solving this JKO step over there. The difficulty arises in two setups. So first of all, we learn our energy by somehow comparing our predictions to the observations in terms of some distributional distance. So how do we backpropagate to the argument? And the even more dominant question is, how do we actually solve the argument in the first place? So there have been several approaches proposed. So I'm skipping this a bit. So one is based on just replacing it on an entropy with an entropy regularized Wasserstein distance. The other one is dependent on the dynamic formulation of Bunne and Grenier. And this is some ideas we are borrowing here, but both of these methods don't scale. And instead, we somehow use the same idea as before. We can replace this Wasserstein distance, or like somehow by the idea that we can find the optimal Morse map by pushing forward our cells with respect to the gradient of some convex function, right? And now we have an argument problem, not anymore over the space of all measures, but over this somehow finding the right or the optimal convex function. And this, in fact, has been in the same 2 weeks discovered by 2 independent works as well. So this is, we are not alone in proposing this. So, but now we have a novel numerical scheme. So now instead of solving this classical JKO step, we are solving the problem where we need to find a convex function, which minimizes the overall objective. So this is an optimization problem in itself. Now we iteratively, at each step, iteratively find the best convex function, which then describes where our population is at the next point in time. The difficulty here is we have now a bi-level optimization problem. On the lower level, we need to find the convex function. On the upper level, our overall goal is to actually learn this potential. And what we can see is that if we look at the cost function to learn our energy functional, this has no direct dependency anymore on the parameters of the energy. So here, the second line, we can see it's only dependent on the optimal convex function, but not anymore on the energy. While, of course, the optimal convex function is dependent on the current parameterization of the energy. But what do we need to do? We need to basically backpropagate through the entire optimization of this convex function in order to learn somehow the energy. And so we do this, and this borrows actually quite some similar ideas from meta-learning or people who have looked at somehow differentiable layers. There's quite some work also by David Duvenant and Zico Kolter, and there was also a neuro tutorial. So they have very similar methods developed in order to learn this in a fully differentiable way. Just to give you a brief overview of why this is useful. So we are in a setting where we want to predict just the flow of a Gaussian along a semicircle. And we are in a setting where we are observing noise over the way. And what is the forward method doing? The forward method, the gradient of this energy directly determines the location of our points at the next step. Meaning it's going to perfectly mimic the noise it sees during training, because this is what it saw during training. We, on the other hand, we somehow differentiate between the push forward operator and the energy itself. So the energy is just learned for whatever the push forward operator is doing. And the push forward operator, if I'm actually at test set observing something noiseless, it's not going to artificially create noise because it's just trying to minimize the energy, or like kind of like move in the direction of the minimal energy which we learned. And so we are getting somehow more stable signals over time. And this is in particular dominant if I'm increasing the noise level, the forward method is really dominant. This is only like a small experiment because this method was more technical, but here we have a data set where we have embryonic stem cells which are developing over time into different like mesoderm, endoderm, ectoderm. And we have different days. So here just the two principal components for visualization. We observe it at day 1, 6, 12, 18, and 24. And we have some form of somehow classification what the cells' identities are. And what we can see is that somehow JKO-net matches this overall trend in evolution, even over like several steps in time. And we are able to somehow capture the heterogeneity of the cell types. So what is next? And this is the last 2 slides with which I want to end my talk. So we have looked at fairly simple setups where we just learned the matching, and this is even complicated in itself. And now with JKO-net, we have entered a bit the domain of like learning flows over several snapshots, parameterized or like kind of learning the solution to some form, like some partial differential equations. There's also a very powerful class of stochastic, like kind of learning systems using stochastic difference equations, which can come in quite handy in future to learn these cellular dynamics. And this all started with like score-based generative models, which are generative models which try to like learn from a Gaussian to generate very high quality images. And the idea here is that starting from images, if I continuously add noise, at some point I will end up at something very random. Then I have a forward SDE, which I can reverse, which gives me some more complicated terms. But this ideally should get me back, if I know the score function, this should get me back into the data space. And this I can now use as a generative model to produce high quality images. Of course, we are not interested in images. It has been done quite extensively currently, or used quite extensively for molecule generation, and so on and so forth. We are, of course, still interested in cells. And we are in particular interested in having 2 complicated distributions left and right. We want to learn cellular transitions starting from some control distribution. And now this doesn't work anymore, this forward SDE. So in fact, we need to replace it with a system dependent on 2 score functions. So we have a score function on the bottom and at the top. And this is work also by De Bortoli and Chen at Georgia Tech, and Valentin is at ENS. And this is actually, this system is known as a Schrödinger bridge, a very famous concept originating from physics, particle physics by Erwin Schrödinger, 1931. And in fact, it is known to be identical, the solution of the Schrödinger bridge, where we are trying to find just like a stochastic, like a system of stochastic difference equations, like the stochastic law, which minimizes the problem. It's actually identical, the solution to entropy regularized OT. So it doesn't matter. All you somehow, the point I want to make is that this is still, in a sense, some form of optimal transport prior on the process. The problem is you need to always choose some reference measure with which you compare the stochastic system you learn to. And it's not really clear what reference process to choose. And some initial work we looked at is that we thought, okay, can we find some data-informed prior, some data-informed reference to make the system easier to learn these 2 score networks better, which somehow allow us to model these cellular transitions. And one approximations we derived is by just saying, okay, I can find a Gaussian approximation of my perturbed and unperturbed states. And now we derive a closed form for this process, for this stochastic process. And this closed form now serves as a reference process to somehow feed into the system of stochastic difference equations, which at the beginning of training already gives us some certain correct behavior, some right directions in which these stochastic difference equations are getting used. So I would like to end the talk with just like a quick outlook of other work we looked at, which is equally exciting, but I didn't have the time to work on it or to talk about it today. And this is where we just predicted somehow retrosynthesis path for drug or for all types of molecules. We were looking quite a bit in the direction of protein design, equally important for drug development, where we would like to find a good representations of our quite complex proteins to learn with. Or I was recently working with Octavian at MIT to look at rigid docking for proteins in eye. And with this, I would like to end my talk and acknowledge my co-authors, because there was a lot of work presented with a lot of people involved. So Cellular was done in collaboration with Gunnar Rätsch and Lukas Pelkmann's group, in particular, my joint first authors, Stefan Stark and Gabriele Gut. The extensions to Conrad was done with my supervisors, and recently the extension to the cell death with Friederike Lübeck and David Alvarez-Melis. Then Yaping and Ivy worked on GSB flow, and Letizia was also involved with JKONet. And of course, my fantastic supervisors doing my PhD, Andreas Krause and Marco Futuri. And my PhD is funded by the NCC Architecture in Switzerland.